Nick, you see my screen, right?
Yep, sure can.
Okay.
It's my pleasure to introduce our second speaker of the day, Cheyenne. She's a PhD candidate at West Virginia University, working with doctors Pat Mazik and Vicki Blazer. Her main project involves investigating immune function in wild smallmouth bass and dealing with the complexities that come along with studying a non-model species in the wild. She works for the USGS Eastern Ecological Science Center at the Leetown Research Laboratory in Kearneysville, West Virginia. And in her spare time, she enjoys training her dog and doing yoga. So thanks a lot for joining us, Cheyenne, and you can take it away.
All right, thank you, Nick. So today I'll be talking about some of the approaches and difficulties that we've run into actually understanding immune response and disease resistance in wild fish. So since 2016, we've been looking at immune response in wild fish and specifically smallmouth bass in the Chesapeake Bay watershed. And we've been interested in this because there have been almost 2 decades of disease and mortality investigations with wild smallmouth bass, and there's been no single or consistent findings as of yet. So they faced a flurry of disease issues, including visible lesions like these red eroded areas here, these red, like this raised mucoid lesion, multiple bacterial infections, heavy parasite loads, like here's parasites in the spleen tissue, fungal and viral infections, including largemouth bass virus, and even the more recently discovered adenomvirus, which has been present in blotchy bass syndrome, which is this picture up here, melanosis of the skin. And so because there are so many stressors that are occurring in the populations of wild fish, we believe immunosuppression may be occurring. And so when you have all of these stressors occurring, like water quality factors such as nutrients and temperature, pathogens and parasites, contaminants, maybe from pharmaceuticals, farms, factories, wastewater treatment plants, we have genetic changes and then storm events that cause high flow and runoff, and then complex interactions between all of these different things. And then it becomes very difficult to pinpoint any specific cause for disease or death that these fish might be experiencing. So in order to test the immune function, our approach has been to develop 3 different in vitro assays with isolated leukocytes from anterior kidney tissue in wild fish. 2 of the assays measure the innate immune response, and 1 measures adaptive immune response. And one of the biggest challenges for testing immune function in wild fish is that the majority of them are non-model species. So this makes it really hard to develop methods for them when you want to label specific cells because commercial products aren't readily available. So for instance, for our work, we wanted to distinguish T and B cells in one of the assays. So we had to spend a lot of time actually working with colleagues at VIMS, Virginia Institute of Marine Science, to develop an anti-smallmouth bass IgM monoclonal antibody specific to B cells so we could label the B cells. So just a quick review, the innate immune response is nonspecific, it acts fast. And then just like in humans, fish have physical, humoral, and cellular defenses as part of this. And phagocytosis, like you see in the video, here is a big component of the innate response. And there are professional phagocytes called antigen-presenting cells, and these are a major link between the innate and adaptive immune response. In the adaptive immune response, it acts on specific pathogens and takes longer to mount a significant response. Sometimes in fish, it can take up to several weeks compared to maybe only 4 to 7 days in humans. And because this response is specific, it creates specific antibodies like this video, and it's capable of remembering pathogens for faster response in the future. So this production of antibodies can be activated by helper T cells, part of the adaptive immune response, or directly from complement in the innate immune response. So just to talk about some of the methods and how this all plays out, the overall workflow for looking at immune function begins in the field and ends in the lab. So in the field, we attempt to collect 20 adult fish at each site, and sometimes we're also able to collect water for analyzing water chemistry. Once the fish are collected, we necropsy them on the site. The anterior kidney is aseptically removed and homogenized into a single cell suspension, which then is taken back to the lab for next day processing. And then the leukocytes from that single cell suspension are isolated in the lab and run through 3 immune function tests. 2 of the assays actually utilize imaging flow cytometry, which is this bottom picture down here. And we have been able to combine machine learning technology to actually distinguish cell types based on morphology, since cell surface markers aren't readily available for our fish species and most wild fish species. The 3 immune function tests we are performing were chosen to test different aspects of innate or adaptive immunity. So we developed a bactericidal killing assay, which challenges the cells with 2 species of bacteria, and we are using Aeromonas sobraya, which has actually been isolated from wild smallmouth bass at some of our sites. And then we're also using Yersinia ruckeri, which is a cold water species that they are unlikely to have been exposed to, although more recently we have seen responses to this at one site, which might indicate these fish have previous exposure. But the second assay is respiratory burst, which measures another aspect of innate immunity, the general reactive oxygen species present in phagocytic cells. And that's the dark red that you see lit up here in the cell. And then the third assay, mitogenesis, is measuring the ability of T and B cells to respond when exposed to 3 different stimulants in the lab. And we are using CON-A, concanavalin A, PHAP, and LPS, lipopolysaccharide. And this assay also involves the IgM monoclonal antibody that I mentioned earlier to label the proliferating B cells and separate those from the T cells. So now let me dive into some of the examples of what we're actually seeing and why it's so hard to make sense of everything. So here's some data from the bactericidal activity, and this is from 2018. And so I just want to point out that we're seeing seasonal differences, and it varies by site. So here at the bottom, we have the site. On the side axis, we have the percent killing of the bacteria by the phagocytes, and in the different colors, we have the seasons. So we have red, which is spring, and blue, which is fall. And so what we're seeing is over here in the spring, we have Pine Creek and South Branch of Potomac. These are 2 sites where the bactericidal activity is actually higher in the spring, the orange balls, than the fall. But then over here, we have Antietam Creek, where we're actually seeing higher bactericidal activity in the fall, the blue bar, compared to the spring. And so it's really hard to pull out exactly what this means, because there are just so many different things that can influence it. One of these things, though, could be land use. So Antietam Creek, which I'll show you later, is more highly agricultural and developed than these other 2 sites. Pine Creek is actually the most forested, and South Branch is highly agricultural, so it's kind of surprising that it groups with Pine Creek. But it's not as developed as Antietam. So then if we want to combine and we want to look at the bactericidal and the ROS production together, because they're related, we are seeing positive correlations between the 2, but only at the reference site Pine Creek. So that's what's highlighted in yellow. And down on the bottom, we have the unstimulated ROS, which is the levels from the cells that are just collected from the field, no stimulation in the lab. And then we have the bactericidal killing activity over here on the y-axis. And we're really only seeing this correlation at Pine Creek, which is the most forested site and actually is our reference site. And we're seeing weaker correlations or none at all with other sites which are more agricultural and developed. So the left is the Antietam on all of these graphs, and the right is South Branch on all of these graphs. And the positive relationship, that's actually what we want to see, because it's characteristic of normal functioning phagocytes, because one function of ROS, right, in phagocytes is to help kill the engulfed bacteria, just like the video at the top. So when we're looking at respiratory burst activity, we are noticing a general trend that during seasons where so if we look on the left graph is the unstimulated reactive oxygen species. This is the level of reactive oxygen species that is the background level. So these cells are straight from the fish, not stimulated with anything. And this is the percentage of the cells that have ROS positive. And what we're seeing is that the respiratory burst and the ROS are opposite. So the respiratory burst is actually when they're stimulated. So anything above this line at 1 means that those cells actually were able to mount a response and create more reactive oxygen species when exposed in the lab. So that is a good thing. It shows that those cells are mounting a response. And what we're seeing is that during seasons where ROS is elevated, like here in the spring, it is decreased for respiratory burst is suppressed in the same season and vice versa. So this is actually expected because too much ROS in a cell can actually damage it. So meaning if ROS is already too high in the cells, then they won't be effective at producing more when stimulated. And that's a defense mechanism and part of the checks and balances of a healthy immune response. And then we have our mitogenesis assay, our third immune function test. This graph is over here showing the percentage of the IgM positive cells, a major population of B cells, at different sites down here for the different mitogens. So there's 3 mitogens here, and then our negative, which are not stimulated, our controls. And it's pretty consistent for looking at just the IgM positive cells that we are seeing most active in the spring and little to no IgM positive activity in the fall. And this makes sense because it's normal to have an increase in antibody levels with an increase in water temperature, which usually occurs in the spring. And finally, this graph is just to show that results can be highly variable, not only within a season, not only between years, but also within a site. And we need to consider what else is going on in the individual. So here we have, these are individual fish on the bottom for 3 different sites. This is 2019 data, and over here on the left side is the mitogenesis response. So basically anything, it's the same as the respiratory burst, it's a ratio. So anything above 1 actually means that that fish mounted a response when stimulated in the lab. So anything above 1 means that fish mounted a response. And you just see that it's just highly variable within one site. And the different colors are the different seasons. So the red is spring again and the blue is fall again. And just because there's so many different, there's so much variance in a site, that's why we are also looking at histology, and we are starting to go back and examine PFAS concentrations, so perfluorinated compounds of forever chemicals in plasma that we've archived. We're also looking at gene expression of immune and contaminant related genes. And yes, so we need to consider what else is going on in the fish, and we need to put all of this together and make sense of it. And that's what's really hard. So what does all of these results mean in terms of immunity? This is just me when trying to make sense of all of this data and put it into context. But the short answer is it's very, very complicated. And I say that all the time whenever someone asks me what a result means. And it's funny because I read a joke that Princeton professor Jessica Metcalf told, and that it was an immunologist and a cardiologist, they're kidnapped. The kidnappers are going to shoot one, but promise to spare whoever has made the greater contribution to humanity. So the cardiologist says, well, I've identified drugs that have saved the lives of millions of people. Impressed, the kidnappers turn to the immunologist and they ask, well, what have you done? And the immunologist says, well, the thing is, the immune system is very complicated. And then the cardiologist says, just shoot me now. And that just resonated so much with me because I say that all the time. And here's why. So there are many, many factors that can influence the immune response, especially in wild fish, as I pointed out earlier. And these are some examples of the data and how it might affect. Here, for one, we have chemicals, and we're having yearly differences. These are 2 different herbicides, atrazine and metolachlor, and the different colors are the years, and the y-axis is the concentration, and the x-axis is the month. And we're seeing yearly differences, but we're also seeing seasonal differences. So we're seeing peaks here at the late spring, early summer, which could correlate to some of the seasonal differences we're seeing in the immune response. And then another thing that could be affecting it is pathogens and parasites. So here's 3 different tests. This one is a mitogenesis to lipopolysaccharide, LPS. And we're seeing a decrease in that response in spring, but not fall, as parasite rating goes up. So these are parasite ratings at the bottom. So as the parasite in the spring, as the parasite ratings are going up, we're seeing a decrease in the LPS stimulated mitogenesis. Same thing for bactericidal activity to Yersinia rucri. Here's parasite ratings going up. And here is the killing to Yersinia rucri going down in the spring, but not the fall. So spring is blue and fall is orange in this graph. Then we have bactericidal activity. And so for bactericidal activity, I had ratings for parasites in 3 different tissues: liver, posterior kidney, and spleen. And in all 3 of those tissues, the parasite ratings correlated and made the bactericidal activity decrease. So the parasite rating is increasing on the bottom, and the killing towards Aeromonas sobriae is decreasing in the fish. And so, and also, as I said, I was going to show you land use, because that also seems to have an effect. Here's Antietam, which I showed you, and all of the yellow is the agricultural and red is developed, green is forested. So there's Antietam, and then we have the South Branch, which has some agriculture, and then Pine, which has some agriculture, but is more forested than the other 2 and has less anthropogenic input, which is why it is considered kind of like our reference site. And so in summary, there are just a multitude of confounding factors that we need to consider. And we know that factors may relate to differences include land use and chemical inputs like contaminants, perfluorinated compounds, water quality factors like nutrients and temperature. Those can also affect bacterial and parasite populations. Pre-existing infections in individual fish play a role, maybe in individual differences. And then those individual differences like age, sex, health status, and even gene expression of immune-related genes is something that we are starting to look at now more. And so we are developing a code set now so we can look at the abundance of genes related to immunity, stress, metabolism, and contaminants. And we are also looking more at histology for presence of parasites, pathogens, inflammation, and other indicators of oxidative stress. And then I forgot to put it here, but also we're going back and looking at the PFAS concentrations in plasma that we have archived from fish. So why is immune function important to incorporate into studies on wild fish health? 1, disease and death historically not caused by one single or consistent thing. There's so many things that can affect in the field that it's hard to pinpoint. 2, the ability to long-term monitor gives a baseline of wild fish, since immune function could drastically be different among individuals and sampling sites. 3, most studies on fish immune function are lab studies and using concentrations that usually are higher than what we see in the field. And then 4, the ability to collect other tissues gives the immune results context and makes them more understandable. Well, that's the goal anyway, but it's proved to be very difficult. And so I just want to thank my funding sources and everybody in the field, the West Virginia DNR, Maryland DNR, Pennsylvania DEP, who help with field collections, because this would not be possible without them. And then everybody who helps with field sampling at Leetown, also all my advisors at West Virginia University. And thank you. And I can take questions if I have time. And I do.
Yeah, you sure do, Cheyenne. Excellent presentation. Complicated indeed, but you did a good job walking us through it. Feel free to unmute yourself and ask your question directly or drop it in the chat box.
Yeah, this is Isaiah here again. That was a really great presentation. Thank you so much. I saw that you mentioned PFAS and had a star next to it, so I take it that you have some interest in that. I'm curious. I don't really know much about PFAS chemical impacts on fish health. I guess you're talking about things from an immunological standpoint. So, what sort of impacts do PFAS chemicals have on immune status in fish?
Well, I think it goes hand in hand with like endocrine disruption, because PFAS, I think it has PFAS has a lot of the same effects in fish as it does in humans. So endocrine disruption, which also plays into the immune response, which then plays into so many other things. But I think the main thing is the immune response and endocrine disruption in fish, just like in humans. And I see that I can send you some more resources and publications, some things that I've been reading about it. I'll copy and save your email address right here.
I would love to have that distillation. That would be awesome. Thank you.
Yeah, yep, no problem.
Oh, also, how difficult was it to get these assays running from, like, what's like the timeline from collecting head kidney samples from wild fish to running this assay in a lab, like probably the mitogenic one.
It takes 4 days for all the assays. So collect the fish, then the next day run some assays, then they have to wait overnight. And then I'll do stuff the next day, and then they wait overnight, and then I do stuff the next day. And then finally on the 4th day, I can read it. So for each site, it takes me a whole week to run all the assays.
Yeah, this is really interesting because we're trying to step up our PFAS sampling in the state of Minnesota, and we're collecting data on that. But a lot of that data, is being used by Department of Health to make recommendations about whether or not to eat fish from those lakes. But there's nothing going on really in terms of connecting with researchers to understand how those data might inform something about fish health. So I'm pretty curious about this. I'd love to stay in contact about it.
Yeah, definitely. Definitely. Thank you. Good questions.
Hi, Cheyenne. This is Gary Marty. Question on the one with the spleen and the parasites. Or I think the parasites kind of went down, I think, in the springtime. I wonder if you've considered the option that the reason the fish have so many parasites is their immune function is somewhat decreased.
Yeah, totally. So it's kind of like the chicken or the egg thing. What came first, like the parasites or the immune function, right?
And I don't know if there's any way to figure that out.
Honestly, that's where I'm stuck at. All of this and how to figure out what it actually is causing what, or what's actually happening first. Yeah, that's where I'm stuck at.
So if you figure it out, let me know. That'll be great.
Definitely, I will. I'll do a presentation when I figure this out.
All right, any other questions out there?
Yeah, one more actually. I'm just curious. I know you mentioned melanistic bass, and I'm curious if I know you have a lot of your data correlated by site, but do you have like enough data to see if there's a link between the melanistic appearance of fish and any immune dysfunction?
So that isn't my project. Luke Iwanowitz actually has been working on that in our lab. And some of my fish, I don't think I have enough fish in my data that actually had those melanistic spots to actually do that, to actually figure that out.
Okay. Yep.
All right. Well, thank you very much, Cheyenne. Great presentation. Very well done.
Thank you.